Okay, thank you, Chair, for the introduction. So, good morning, everyone. My name is Zhaojun Zhang. Okay. So today I'm going to present our RNA-Skim paper. I'm a PhD student from UNC, and my advisor is Wei Wang, who's sitting over there, and she's also the second and last author on this paper. So before we start talking about the RNA-Skim, I want to briefly review the existing work on RNA-Seq quantification. So we categorize the existing methods into two groups. The first group we call it's called alignment-dependent methods, and it has been developed for several years. So this type of method typically require a liner to align reads back to the genome or to the transcriptome in order to figure out their original transcripts. And the alignment problem is typically very computationally intensive, so it typically takes hours, if not days, to finish, even for analyzing a single sample. And so here is a very simple illustration. So here is a transcript, and so all the reads align to this transcript. And so once we have this alignment result, the next step is to estimate abundance level for this transcript. And so here is not a very perfect description of what happened behind this model, but we think it's very helpful for us to think about this and come up with the idea about RNA-Skim. So what we think about this model is like people typically will first generate this black curve here. So each point on this black curve represents the number of reads that covers the corresponding base pair. And after this model generates this black curve, actually all the detailed alignment information actually can be discarded, and only the size of area under this black curve will become the sufficient information we need to estimate the gene expression level or abundance level for this transcript. So this is the first group. So the second group, we call them alignment-free method, and it's actually recently came up a Zhaojun paper. I think it's right now there's only one method there, and the RNA-Skim is the second one, to best of our knowledge. So for Zhaojun, the quantification part is actually very similar with the existing with the alignment-dependent method. But the Zhaojun does not use alignment information. Zhaojun uses k-mers to quantify, use the information about k-mers to quantify the transcriptome. And because it successfully gets rid of the alignment step, so it actually can give us a very huge speed up. It's 10 times speed up comparing to the alignment-dependent methods, but still provide comparable results. So why do we want to build this RNA-Skim? We should focus on the speed. So we think this figure gives us enough motivation. So this figure shows the gap between the growth of the data size from the sequencing, and the improvement on the computing infrastructure is actually increasing exponentially. And if this trend continues, so one day, and we do not have improvement on the software level, so one day all the valuable information we can generate from the sequencing technology won't be able to analyze it by the existing software. So we think that will become a sad thing, and we think this is actually a very practical challenge. It's not just some theoretical question. So here's why. So, but how? So let's go back to this black curve. So we think existing methods, no matter it's alignment-free or alignment-dependent method, it's always try to integrate, calculate this area by integrating this area base pair by base pair. And so how can we? So the question is, can we approximate? Can we have approximation to estimate this area? And from some basic concept from numerical computing, we know that we can actually just sample a subset of base locations and use their hats to get another line which approximates this black line, and then we can use this blue area to approximate the area under the black curve. So why does this work? So two reasons. We think first, RNA-Skim data itself contains noise, and errors from sequencing from sequencing part. And secondly, we think that abundance level of transcripts are actually random variables. If we run the same experiment repeatedly for the same sample on the same condition, we typically will observe different values, but they will follow the same distribution. And so we think as long as the error of this approximation introduced is reasonably small, say a magnitude smaller than the variance of the transcript level, then we think that our estimation will be still comparable to the existing work. So based on this idea, we came up with a concept called sigmers. So sigmers is so we first depend on the definition of a partition of a transcriptome, which we just partition transcripts into different groups. And sigmer is actually a chimer that occurs in only one cluster. So each sigmer actually uniquely identifies containing cluster. So here's the example. It's a transcriptome of 5 transcripts. And we put the first 3 transcripts into one cluster, the last 2 into the second cluster. And here are 5 sigmers. I color them. They are not exhaustive list of all sigmers in this example, but we think a fraction of sigmers should be enough for us to quantify this transcriptome. So for example, GGAA is a sigmer, and because even though it occurs in 3 different transcripts, but all 3 transcripts belong to the same cluster. And the TC is not a sigmer because it also occurs in the second cluster. So it's a new concept, but we want to have some similarity with the existing concept. So we actually think sigmers are just special reads. They are actually reads. So why do we think about that? So here's the same example. And when we do the RNA-Seq, we typically will get a list of reads. And some read will cover some sigmers, some read won't cover any sigmer, that's fine. And then we detect all the sigmers in the reads, and we generate this list of sigmer information. So when we look back to think about how this data is generated, we can actually omit the intermediate step, and we think that these sigmers are directly generated from the transcriptome. And based on this, all the gray areas can be ignored in the transcriptome, and each transcript is instead of represented by a string, it's represented by a set of chimers. So that's why we think sigmers are special reads. So based on this data, how do we quantify the transcriptome? So we propose a probability called alphas. So alpha is the probability to represent the probability of sequencing a sigmer from a transcript. And so based on alphas, we can calculate the likelihood of how a sigmer data set is generated. And I do not have time to go to detail, but the high-level principle is that we try to maximize the likelihood of all observed sigmers in this data. And so just want to remind you, in the alignment-dependent methods, they want to the principle of their estimation is to maximize the likelihood of observed alignments. And for the sailfish, a sailfish tries to maximize the likelihood of all the observed sigmers. Actually, all 3 types of methods, they have exactly the same mathematical model, but they just interpret the variable differently and use different type of data to quantify the transcriptome. So here's the overview of RNA-Skim pipeline. So we have a transcriptome, and we go through RNA-Seq, we have a list of reads. And the first step is only based on the input from the transcriptome, which we cluster the transcripts into different groups based on their similarities. And then we detect all the sigmers, and we select a subset of sigmers from there. And the third step is to use this sigmer information and to detect all the selected sigmers in the RNA-Seq data, and then we will quantify this transcriptome by estimating those alphas, by maximizing the likelihood of observing all sigmers. So in addition to this, I want to talk about 2 optimizations we did in order to make sure the performance is optimal. The first optimization we did is how do we find all sigmers. So again, here's the definition of a sigmer. So it's actually a very simple programming problem, and I believe everyone who have some basic programming skills can give me a program like within 2 hours. So the straightforward implementation is actually quite easy, but in order to do that, we need to store all the chimers in the memory, which right now for the human genome or for the mouse genome typically require tens of gigabytes of memory. So instead of directly storing all the keywords in the memory, we use Bloom filters to reduce memory usage. And Bloom filter is a probabilistic structure to represent a set of elements. And actually, no real element is stored in the memory, and it's provide a very efficient add and lookup operation. So in our implementation, we take advantage of this property in Bloom filter, and the RNA-Skim always report valid sigmas, but sometimes it may miss some valid sigma. We think it's fine because we don't use all the sigmas, we only use a fraction of them. So the second optimization we did is to optimize the procedure to detect all the sigmas in the RNA-Seq data. So here's the problem. We have a list of sigmas and want to detect whether how they occur in this set of data. And this is actually still a very old computer science problem. So it's called a multiple pattern searching problem. And a straightforward implementation will be OKN. K is the length of the sigmas, and N is the size of data. So what we do is we use this it's called the Robin-Karp algorithm, or call it a rolling hash algorithm, which provides a linear complexity. So, and we successfully get rid of K in the complexity, the big O complexity notation. So which means our algorithm is actually invariant to the size of sigmas. We think it's very good because for the existing element algorithm, it's always linear to the length of the read, and the length of read is getting longer and longer. But if we want to use sigmas, it's actually even though we can try different lengths of sigmas, but it still gives us the same running time, and we think it's a very nice property. So how do we do that? So here is a read. And we want to check every formers for the keymers with length of 4, whether they are a sigma or not. So for the first time, we'll check the first former, and it's a regular check to calculate the hash value and look up this in the hash set to see whether it exists in the existing keymer list or not. And for the second one, because it's just next to the first one, and actually it's only we pop the first character out, and then we append the last character in, so there are actually some overlap areas. So when we calculate the hash value, we can actually reuse some existing information, which means that the second one, the hash value is actually calculated in a constant time instead of instead of OK complexity. And we continue to do that for the remaining formers, and till the end, then basically our complexity will become the length of this read. And so we think this which is very nice. So here again, this is the RNA-Skim pipeline. We have transcriptome, generate RNA-Seq data, and the first one, then we calculate all the sigmas, and we detect the sigmas in the read data, and we quantify them through some EM approach. So here I want to show you some simulated results. So all the simulated results are based on, and the real results in this talk are based on mouse population. And we use Flux Simulator to simulate one sample with 40 millions of 100 base pair arrays. And the y-axis here represents the ground truth, and the x-axis represents the estimation from RNA-Skim, and each point here represents a transcript. So ideally, we want to see a diagonal line, but because there are some noise in the data and some variance in the estimation method, we'll see this, but we still can see this clearly linear trend. So we also tested under 100 different samples, and so we compare our method with the other 4 methods. We compare it with Sailfish, Cufflinks, RCM, and Express. So we will apply these methods and compare them under 4 different metrics. 2 are correlations, Pearson correlation and rank correlation. 2 are rates, false positive rates and false negative rates. And here's the result. So for the best performance method, I put it in the board in a board font, and we can see that actually the message here is that a single method dominates the other methods across all 4 metrics. Actually, for these 4 metrics, we see 4 different methods become the first one. So, but the other thing is that instead of difference, we want to emphasize the similarity. So these methods actually show very comparable correlations for the first 2 columns, and they show some interesting trade-offs on false positive and false negative rates. For example, for Sailfish and Express, we can see it reports relatively high false positive rate, but relatively low false negative rate. But for the other 3, it typically report a relatively low false positive rate, but relatively high false negative rate. So not a single method win, but we think this is good because at least RNA-Skim didn't do anything terribly wrong compared to the other 4 methods. So instead of just work on the simulated data, we also tested on the real data. And for the real data, we collected both RNA-Seq data and the microarray data from a population with 54 inbred F1 mice derived from 3 different founders. And here I want to emphasize that microarray data is not ground truth. The trick about this type of experiment is that we don't know the truth on the real data. So we really couldn't do anything more than this. And so here's the result. Still the 5 methods, and we test the correlation between the estimated value from RNA-Seq data and the estimated value from the gene expression data. And we can see that they actually have a relatively same correlation level, and we think which is good. Basically, it means that RNA-Skim still provide comparable results to the other 4 alternative methods. Okay, so why do I spend so much time to tell you that RNA-Skim does not do anything better than the other 4 methods? So here's the reason. So because RNA-Skim is faster, so we tested on a sample with 44 millions of paired reads. And so here is the CPU time, required CPU time, and RNA-Skim can be require 10 times less CPU resources than Sailfish, and require more than 100 times less CPU resources than the alignment-dependent methods. So in conclusion, I think that the redundancy is very important in RNA-Seq-based analysis, and it really should be considered in the statistical modeling and algorithm design. And to best of our knowledge, RNA-Skim is the first one to use to take advantage of redundancy in the RNA-Seq quantification to solve the RNA-Seq quantification problem. And as a proof of concept, we propose sigmas, which can be used as a novel presentation of transcripts, and we develop RNA-Skim as a lightweight application to prove this approach really works. So in the end, RNA-Skim is actually open source software, and you can download it from this link. And it includes 5 very small and independent components. So if you are only interested in the counting part, you can just get that from the software and embed that into your research. And hopefully that will be helpful for your research. And I think that ends my talk. Thank you very much.
So we have time for questions. There's a microphone in the middle of the aisle here for questions.
So that was a really great talk. Thank you.
Thank you.
Quick question though. Low abundance transcripts. Where do they end up in the clustering, and does that affect your ability to quantify low abundance transcripts? Because I can easily believe that your method is doing amazingly well on large abundance. Yeah? So could you talk a little bit about the case for lower abundance transcripts, please?
Sure. So I think low abundance transcripts is very hard to solve, and even if you want to use alignment-dependent methods to align the reads to, because we only will observe a few alignments to the low abundance transcripts, which makes it really hard to solve. So, but this plot here shows that even for the low abundance transcripts, we still perform reasonably good. And from the overview correlation scores, we think we are just doing fine as well as the other methods. So, yeah.
When you do the EM to optimize the alphas, the relative frequencies, what happens when it's a degenerate solution, or when there are lots of different alphas in the space that can actually maximize the likelihood? Have you considered what happens in that case?
then
Or if even slightly different might
Yeah.
suboptimally maximize the likelihood?
I think we need to, either you can penalize alpha to give some prior information on the alpha or do other things. But in addition to that, because the data didn't show any information on whether this sigma belongs to this transcript or the other transcript, so we don't have any solution right now to do that.
Thank you for the great talk.
Yeah, very nice work. I have two questions. The first one, how do you deal with the multiple mapping reads?
So the sigmers you still have like a similar problem. So for the sigmers, it will belong to multiple transcripts, and the information will be, it's the same approach in the alignment methods. So we just, so through the EM, we can redistribute the weight of sigmers to different transcripts and to disambiguize the probability. But it's the quantification model is the same with the other methods. So if you So what you would do for the multiple alignment problem, you still want to do the same thing in the sigma, and that's what exactly what we did.
Related question is how do you deal with the feature overlap? Let's say the gene overlap or the transcript overlap. I mean the same region could corresponding to different gene or transcript. How do you assign the rates in this situation? Yes.
So we first cluster the transcripts based on their similarities, not based on their locations. So if there are two genes, they are either overlapped or they share some similar sequences, we'll put them into one cluster, and then we select sigmers based on that. So it's very likely to happen that some sigma can be only assigned to a unique transcripts, and some sigmers can be assigned to multiple transcripts. And then based on that, we use EM model to address the ambiguities among the sigmers that's assigned to multiple transcripts.
Any more questions? Yes, please.
I have a question not necessarily related to your method, but in your comparison with the other methods, you use the, yes, over there. The last two, I was wondering whether you chose bow tie over other splice-aware alignment tools, because the bow tie doesn't.
Oh, we align the reads to the transcriptome instead of genomes, and that actually got rid of this to detect the splice junction problem.
So what was that reference?
The reference genome is generated from B6, but we test it under we test our data. So we use B6 as a reference genome, but all the simulated data are generated from Casca, PWK, and WSB as a reference genome.
All right.
Okay.
Thanks.
Any more questions? If not, let's thank the speaker again.